All right, so in this video, we're going to be starting the introduction of chapter 16, which is on DNA and DNA replication. And the majority of what you guys are going to have to know is going to be the enzymes and how and when and why it would need to replicate. But notice what this chapter is actually titled. It's the basis of inheritance, so how things are actually passed down. Now, there's debate in terms of who actually discovered DNA, Watson and Crick or Rosalind Franklin. We'll save that for another video or talk about it in class. But the one thing that I need to focus in on is how do you know that DNA is going to be the genetic material? And a lot of AP test questions, sometimes they're going to take the concept of what DNA is and kind of throw you curveball questions. So it's important not so much to memorize who discovered what, but at least kind of understand the theory behind some key experiments. So it's interesting because up until 1950, we really were still kind of unsure of what the genetic material was, like what gets passed down. People thought it was protein, some thought it was the DNA. And one big experiment that we need to make sure that you guys feel okay with is this Meselson-Stahl experiment. So what that concluded. So basically, they're going to talk about the heavy versus light nitrogen. And so I'm actually going to show you the Bozeman clip for a few minutes where he explains what it is, and then we can kind of continue on. I just want you to pay attention to what he's saying because he does a really good overview of what the Meselson-Stahl experiment is. So just watch this part, and then we'll talk about it right after.
So what did they do? Well, basically, this is our Escherichia coli bacteria. They grew in heavy nitrogen. What does that mean? Well, nitrogen normally has atomic or mass of 7 but heavy nitrogen spin it in an ultracentrifuge. DNA that contains this heavy nitrogen is going to be on the bottom, and those that contain lighter nitrogen, they have look at the density of the DNA, figure out what nitrogen is actually being there. So if you think about it this way, thought experiment. Let's say we started with heavy nitrogen, the bacteria make copies of themselves, unset that DNA in the middle, just in nitrogen. Each of those are going to be all heavy nitrogen. They're the copy of themselves. And from a line, they 100% of them are nitrogen. So what they did is they brought them in there until all and switched it to people living in the lighter normal. So basically, the lighter nitrogen in the simulation. So bacteria generation 0, we'll say, are going to be 100%. They unset and make copies of themselves. So this 100% heavy nitrogen. This first generation, we're going to have 100% of this hydrogen. We're then going to go through another copying process. At this point, we've got one hydrogen, another light strand, and a hydrogen strand. So at this point, it would be 50%. If the semi-conservative theory is right, 50% light nitrogen. Now think in your head if we were to do one more combination. Now we have a 75. So that's what the semi-conservative predict, and that's what they found. 100% hybrid in the second. And it's the so what did that tell them?
All right, so let's kind of introduce what that experiment confirmed. Now, again, on what they were showing you on that video was a couple of interesting components about how it actually is replicated. So in an attempt to determine how it's replicated, the Meselson and Stahl performed an experiment where nitrogen 15 was used initially, just like that video was explaining. And basically they cultured it, and the N15 just means that it has heavy nitrogen. So then when they grew it in a culture where there's only that nitrogen 14 available, that's where you started to see this separation, that after the first generation, you got this hybrid, half of the heavy nitrogen, half of the light one. When they replicated again, you got that one hybrid, but then you also got the nitrogen 14 on either side. And so basically it just did that confirmation that DNA replication is semi-conservative. So that's one of the key components about how we figured out how DNA is actually replicated. And that's really what the whole chapter is about. So it's kind of a really, really helpful experiment for us to understand that DNA is semi-conservative when it replicates. So before we dive into this, let's kind of at least introduce some of the bigger rules here. So you want to make sure you understand DNA is composed of nucleotides. So remember, there's three parts. There's the sugar, the phosphate, and the base. So remember that here is your sugar, this is your phosphate, and then there would be your base. Okay. Then that's going to be the basics of each nucleotide. Now, what changes is if you look over here, the bases, we have a thymine, we have an adenine, we got a cytosine, and we got a guanine. So these are the four bases that are going to be found in DNA, but notice that the backbone is going to be just the phosphate, the sugar, the phosphate, the sugar, the phosphate, the sugar, the phosphate, the sugar. That's what we're calling the backbone. The bases are what's going to change. Now, as you guys are going to understand, and hopefully you do already a little bit, is that the order, what is the point of DNA? It's just to be read to make a protein. So DNA by itself, it's important, but it's only important if it's actually going to be expressed, meaning you're reading that DNA to make RNA to eventually make a protein. So don't lose track of the bigger picture. So regarding that, if you look here, we have purines versus pyrimidines. Now, what we really have to know, remember purine, I remember pure as gold. So adenine and guanine, if you look here, adenine and your guanine have two ring structures. Now, you're not going to have to draw them, but just know the difference between purines and pyrimidines. So just remember purines are adenine and guanine. Pyrimidines are going to be your thymine and your cytosine. And the reason why that's important is because when these things line up, when you're actually building a complete double helix, it's Chargaff's discovery, where basically the percentage of adenine is always going to match up with the percentage of thymine, right? So if I had 20% of adenine in a DNA strand, I'm going to have 20% of thymine because A pairs with T. And if I had 20% of adenine, 20% of thymine, that makes up 40%. I'm going to then have 30% of cytosine bonded with 30% of guanine. So that's a nice double structure. Now, before we get any further, I just want you guys to make sure that when you look at the bases, like when an A pairs with a T and then a C pairs with a G, the bonds that hold them together are hydrogen bonds. And the reason why that's important is because those rungs, right? So actually holding them together, held together by those hydrogen bonds. It's pretty important to understand that it's a weak-ish bond because DNA has to open up to be read. And so one other key term here is anti-parallel, that one side of the DNA is going to be really upside down in relation to the other. And we'll talk more about the anti-parallel structure in the next video. But just remember being able to identify your 3' and 5' carbon. So regarding this, before we go far into DNA replication, what's the purpose of DNA? We kind of mentioned that earlier. It's to replicate, not so much to replicate, it's to read it to actually make a protein. You have to actually read that DNA to make RNA. RNA is then read to make your protein. That's the main purpose of DNA. What's DNA made up of? It's those three components, the sugar, the phosphate, and the base. Why/when would you need to replicate DNA? A couple of things. So really that's like another question about anytime you do mitosis or meiosis. So why would you need to do mitosis? Really growth and repair. And you also need to replicate your DNA when you do meiosis to make your gametes. So anytime you need to replicate your DNA, just think back to mitosis and meiosis. And this just kind of reminds you of that semi-conservative model. The base pairing exists as DNA strands serve as templates for the new complementary strands, right? So if this was your nitrogen 15, if we go back to the mesosome install, open it, this would be the nitrogen 14. And so that's what that whole semi-conservative model is going to represent, all right? Now, before we get into DNA replication, I'm just going to end it here on this short video just to kind of introduce the mesosome install, semi-conservative, and we will see you in the next video.